The next speaker in the session on soft interface engineering is Paul Clegg from the University of Edinburgh.
So thanks very much to the organisers for inviting me. So this is about materials where they're dominated by fluid-fluid interfaces, okay? And I shouldn't stand too far away. Okay, we're doing fine. I'll stand over here. Okay, so I've got the pre-lunch slot. Okay, so everything is somehow profoundly food-related, although there's plenty of colloids. Okay, so the first half of the talk is basically doing something which was entirely inspired by a foodstuff. Okay, and kind of taking a concept out of food and trying to do it in the lab. And the second half is more or less the opposite way round, and is trying to use something from colloidy soft matter in order to try and solve a problem in the kind of food industry. Okay, so the kind of inspiration was this cake icing, so cake frosting, depending where you come from, which is called the buttercream. Okay, and so the term buttercream covers a multitude of sins, okay, and I have one specific thing in mind here. So you can make them in a variety of ways depending which country you come from, but I'm specifically interested in making an aqueous foam from egg white, and then making an oily foam by melting butter and whipping that up, okay? So you end up with bubbles in an aqueous system, bubbles in an oily system, and then you fold them together to make a soft solid. Okay, so what could possibly go wrong? Okay, so the interest was, can you do this in the lab? Okay, and well, we kind of know from shaving foam and meringue and stuff that if you make a reasonably dry foam, it has a yield stress, okay, so it can stand up. So this is Bernie Bink's kind of particle stabilized aqueous foam, but because it's got a yield stress, it's not immediately obvious that you know, if you start mixing two foams together, they are going to have a bit of a job trying to flow around each other. Okay, so that's the kind of starting point. So then. In doing this, I'm gonna realize this by making particle stabilized oil foams, particle stabilized aqueous foams, and mixing them together. Okay, so this is the underlying idea here is then taking colloidal particles and putting them on a liquid air interface. And so then this is somewhat different to molecular surfactants because the energetics is associated with removing shared area between the two liquids, and you're on a much larger scale compared to molecular surfactants, and so the trapping strength tends to be extremely high. Okay, so it turns out that you can make extremely stable foams by stabilizing them using particles. Okay, I definitely can't stand over here, so let's, okay, back. Okay, so when you do air-water, you have to make the particles somewhat hydrophobic. When you're doing air-oil, you make them oleophobic. So you, like Daeun's silica that he was using in the previous talk, you can take those sort of silica particles and put methyl groups on the surface. To stabilize air-oil interfaces, you use partially fluorinated particles, okay? And this isn't the only thing you can do, okay? People make edible versions of these things with fat crystals and so on. Okay, so these are my base ingredients. So this is my kind of aqueous foam system, and so I'm using silica nanoparticles, and I'm not using pure water, okay? So rather than try and tune up the surface chemistry perfectly, instead I modify the aqueous phase by using various proportions of propylene glycol. And having done this, you find under some conditions you get quite a lot of foam, and it's quite stable. Okay, this doesn't look like whipped egg white, right? So the difference here between the kitchen version and what you do in the lab is it's possible that we're not super good at this, but you tend to have a vast excess of solvent. Okay, so you've got a lot of liquid with a nice foam on top. it turns out that Nile blue will stick to the silica, so you can kind of see that the particles are on the surface in a confocal. You can also use freeze fracture electron microscopy to check what your particles are doing. Okay, so that's the aqueous system. The oil, we're using partially fluorinated industrial particles, so they have a particular code number, and you can mess around with a variety of different oils. We ended up being happiest with olive oil, okay? Possibly we should have taken some more straightforward alkane or something, but olive oil with these particles works very well. The particles are much larger, so you can see them under the confocal, and you can see them after they've been freeze fractured. Okay, but both of these, so I've got a foam, and I'm showing you a picture of a foam, you know, a week or so later. So these foams are pretty stable, okay? And you know, this doesn't look like, obviously it doesn't look like whipped butter, and the other one doesn't look like a meringue, but you've got a huge subphase of liquid, and what you have to do here is you have to kind of harvest the foam off the top of the liquid, because you don't want this stuff, because that's going to destabilize everything. Okay, so that's what we do. So we kind of harvest foam off the surface of the liquid. Working in volume just doesn't work at all. So everything's done by mass, okay? So when you're kind of controlling where you are in phase space, you vary the kind of mass of oil foam and mass of aqueous foam, and then you can shake them together. And these things, they're robust enough that they survive that sort of treatment. Okay, and so having done that, what it turns out you get is something which is basically like an emulsion of two foams, okay? So this is oil droplets in water, and this is water droplets in oil, but both phases have bubbles in, okay? And so you can mess around with this. So we've also done something different, where we drain and mix at the same time by doing everything while spin casting, okay? So you have a spinning plate and put everything on it, and you kind of squirt the liquid out and combine your ingredients, but I'm not doing that here. Okay, so yeah, so this is kind of the kind of full range of behavior. So the green phase here is the aqueous phase, the purple is the oil phase. So you start off with green droplets up here and finish off with purple droplets here, and there's a kind of crossover sort of phase separated region where neither of the phases becomes the continuous phase. But it's pretty stable. So I haven't got all of these compositions on this here, but the phase separated one is unstable, okay? But that's still not completely falling apart in a month. It's not that bad. So this is varying. What we're varying here is the amount of propylene glycol, okay? So we're varying the wettability of the more kind of the particles that are in the aqueous phase, and modifying the system. Okay, so just to kind of compare this to what other people have done in this space, so this is our system of sort of droplets of one foam in another foam background. So Sven Behrens also works with aqueous, oily phases and bubbles, and but his oil phase basically coats the bubbles. Okay, so he has a very small fraction of oil in his sample, and it coats the oil, the particles sit in the oil, and so there's a kind of particle-oil interface rather than the particle-air interface, and you get other effects with surplus oil. Anina Salonen also works with aqueous oil and bubbles. So the bubbles here are these white regions, but essentially you have a bubbles in a emulsion background, okay? And so the kind of difference between this and these is you basically have these two populations of bubbles which are kind of happy in two different phases simultaneously. You don't have a kind of separate bubble phase that does one unique thing. It's kind of two separate media, each with their own bubble population. Okay, so obviously in a cake frosting, people are kind of spreading this stuff over the top of their sponge cake or whatever, and so we needed to look at our flow properties. These things don't survive an infinite amount of abuse, okay? So this is then a kind of stress value at some particular shear rate. as you vary propylene glycol and go across the composition space, the phase-separated systems are weakest. These are the unstable ones. But then if you crank up the shear rate and come back down again, you find the flow properties have changed afterwards. Okay. Yes, so that's, but it's basically G prime dominates G double prime. It's a kind of soft solid system. Okay, so so what I've been showing you here is all controlled by the volume of propylene glycol, and you can do all the same stuff by changing the mass, the proportions of mass of the two foams, and so you can go from water droplets to oil droplets via changing the proportion. There's no phase separated states here, and there's a much more abrupt transition between the two states. Okay, so I've created a lot of interface in this system. Okay, so we've got these kind of air-liquid interfaces, but when you mix these two things together, you're creating all this aqueous oil interface, okay, which is the new thing you've put in there. And so the question is, what happens to the air or oil interface, the water-oil interface in this system? Well, it kind of turns out that these samples are almost designed for this energy dispersive X-ray technique, which you can do with freeze fracture. Okay, so we've got olive oil, we've got an aqueous solvent, we've got silica particles, and we've got fluorinated particles. And so you can more or less distinguish everything in this sample. So the olive oil is kind of light blue, fluorinated a dark blue. The water is more or less orange, and the silica is pink. So obviously there's carbon in other places, there's oxygen in other places, but basically the dominant proportion of each element look like this. So you can freeze fracture your sample, and you can more or less look at what's going on in different regions. And so we take an interface between olive oil and the aqueous solvent, and in various places in this sample. So we did one here which is averaged over a whole interface, and these are spot measurements. This isn't the most convincing result I've ever tried to present, but there's a tiny silica peak, okay, typically at the interface. So this is like you're going from water here, which is blue. The water drops down, there's an interface to the olive oil, and just where there's a crossover between these two solvents, there's a tiny pink peak, and the same here. So this is water to olive oil, and there's a tiny peak here. And so what it seems, I can show you the same thing for another composition, but what it seems like is that you make these aqueous foams, and you mix the whole thing together, and you don't necessarily keep all the particles on the air interface. You end up with a certain proportion ending up stabilizing the liquid-liquid interfaces in these systems. Okay, just as so that was what I wanted to say about this cake frosting type project, but just as a kind of stand back from the details, in soft matter, a lot of us are doing things with more and more ingredients that get more and more complicated, and then you're trying to kind of navigate a vast space of possible samples, and you're kind of wondering, have I found what's interesting about this system? You know, what else is going on here? Obviously, there's computational approaches, you know, so L'Oreal and lots of other people use kind of Gaussian process models to navigate through the space of formulations that they could be making. But an alternative approach to the computational approach is to kind of look around and see what people have been doing in the kitchen or elsewhere for hundreds of years, and look whether that's kind of an interesting point in this multi-dimensional space that could then be fruitful for exploring what you can do with composite materials. Okay, so that was my food inspiring the lab. And the other way round is to take something from soft matter and look inside chocolate, okay? And so this is a project that's kind of funded by the kind of Mondelez BBSRC consortium which funds PhD students across the UK. Okay, and so what Mondelez, which is basically Cadbury's, would like to do is to have, so there's obviously chocolate bars which have air bubbles in, okay, and you remove some volume of your high calorie content by having bubbles, but they're interested in having a different design motif where they remove high calorie content and replace it by water droplets. There's obviously problems here related to the water activity, okay? So in a chocolate bar, you've got tons of sugar as well as lots of other things. And so the water would obviously prefer to migrate out and go and join the sugar and create a kind of sugar solution in the chocolate, and so then I can't go back quickly. So these chocolate bars would then become soggy and disgusting and stuff. So you could obviously solve that problem by shoveling salt or sugar into the water, okay, and changing the water activity. But that's not quite what they want to do. They just want kind of water to get rid of calorie content, and they don't want other stuff which they then which changes the flavor or otherwise of the product, okay. I'm not sure we need this for this audience, but obviously droplets can coalesce. Ostwald ripening is the process where you get liquid movement from small droplets to large droplets because of the surface to volume ratio. But what we're interested in here is compositional ripening, where you get movements more or less from osmotic pressure. So you get a chemical potential difference between two droplets due to the composition difference between them. Okay, so we've used particles on interfaces so far, but the interesting background to this is that a variety of people have shown previously that you can suppress Ostwald ripening by making the surface of a droplet rigid using colloidal particles, okay, and there's kind of mechanical effect and there's a curious interfacial curvature effect, and so yeah, I won't backpedal, it's too painful, but one of the ripening routes can be killed off that way. So Bernie Binks in Hull, and he was working with Firmenich, who's like a perfume manufacturer, and I think a group at Nestle also have investigated using particles on surfaces to kind of kill compositional ripening. And what they all saw was they saw kind of cluster formation, and they kind of decided this wasn't looking good. Okay, so how we're doing this is kind of making a model particle stabilized emulsion, or we actually create two emulsions, and one of them is then pure water droplets, okay, stabilized by PMMA particles. The other emulsion is sugar solution of varying concentrations, and we mix them together and we watch what happens next. Okay, and so we're basically interested in, if you mess around with the interface, what happens to the ripening. Okay, so these are pictures of what we've got here. So there's, we obviously play around with dyes in an effort to see what's going on. The particle's quite large, 700 nanometers or so in size, sterically stabilized PMMA, and we use different dyes for pure water versus sugar solution. Okay. Yeah, so the resulting emulsion is very stable. Okay, you can, some of the tiny things you can't find between these two, but basically they don't change over several days. And that's, so this is what PMMA looks like on the surface of a droplet. So for this, yeah, so I didn't say it on a previous slide. This is a maximally, apart from PMMA, the other reason this system isn't edible is the oil here is dodecane, okay? So this is completely, this is not for lunch, okay? But with the kind of water, dodecane and PMMA, you get a nice coating over the surface of the droplet. Okay, so there it is. Okay, so this movie then is, so we've got pure water droplets in yellow here, and we've got the sugar solution is in purple. Okay, so then this is then a movie over 2 hours. so you can kind of see these water droplets shrinking as the water moves out to the sugar solution, and in places you should be able to see, if I backpedal, you should see coalescence events amongst the sugar solution droplets. There, bingo, that one just went, and that one, there you go. Okay, so the water's being sucked out of the pure water by the concentration gradients of the sugar. We've got a kind of monolayer of particles over the surface, so as the sugar droplets swell, you then open up holes, and so you get these kind of coalescence events, and so this is all fine. Now, the curious thing for dodecane, which other researchers haven't seen previously, is this here. When it gets to the end, it blows up, okay? So over 2 hours, not many of these explode, okay? There's only 1 or 2 explode. If you run this over the weekend, the end fate of all the droplets is finally the particles redisperse, okay? So finally, the sugar is enough to basically pull the interface off the particles, take all the water out, and the droplets, the particles are basically then redispersed and fly away. Okay, this is dodecane. Sorry, what's that? Yeah, so then so in the purple here is we do various concentrations from 10% up to maximum, which is 64% volume of sugar, and the sugar won't come out into the dodecane, and so you get this concentration pulling the water the other way. Yeah, this is varying the sugar concentration. So we go between 10% and 64%, and this is the radius of the pure water droplets versus time over 2 hours. And if you crank up the sugar concentration, the water all moves faster, as you might expect. But there isn't a point where the compositional ripening stops. Okay, so with 10% sugar, it's still the water's still being pulled across. Things are still crumpling as the water is moved into the sugar droplets. It's all just much slower. Okay, so the take-home thing is in dodecane, ripening is not prevented at all, and the cute thing is that the droplets finally explode. Okay, so blah 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 blah. Kind of said that. So this is so previous people have been kind of looking at things crumpling, and they haven't seen this kind of exploding motif. Okay, so that was kind of an oil which is never going to go anywhere near food, and so so we switched to a triglyceride. Okay, and so when you change the oil, everything changes, as you'd expect. So this is our triglyceride, and so one level that you switch to this oil, and it's going to change the solubility of water, it's going to change the kind of diffusion behavior, but it's also going to change the interfacial tension, and it's going to change the particle-particle interactions. Okay, so basically everything changes. So this is our system, which doesn't quite look the same as previously. So one feature of it is that, so we've got these water droplets in the triglyceride oil, and a feature of it is that you also get particle clusters, okay? And so not all the PMMA is fully dispersed. Yeah, so so we've got these clusters, which I'm just kind of drawing attention to. But the other thing is that unlike the dodecane, the coating on each water droplet or sugar droplet is far from perfect. Okay, so there's disorganized partial covering of the droplets with the PMMA. Okay, so evidently the colloidal interactions in this system are quite different. Okay, so we do the same experiment again. So we've got sugar solution is purple, and the water droplets here are black. Okay, and we've the dye situation was different, and we've dyed the continuous phase. Okay, and so you're getting the same game here. I'll maybe play it again in a minute, but if you watch this space, you are seeing these kind of crumpling behavior. Okay, so watch this one here. Okay, it crumples, and you get these kind of messy little lumps of colloidal aggregate, and they're long-lived. Okay, no explosions in this system. You kind of get these crumpled states. Everything is much faster, and the crumpled end state of these particles never they never explode. Okay, so these crumpled lumps of things are the same as the Hull collaboration and Nestle and other people have previously seen when they've looked at compositional ripening. And yeah, you can vary the sugar concentration in the same way as we did previously. What's different here, so this isn't zero here, okay? And so you basically shrink down to you hit a plateau of a final water droplet cluster, which has a fixed size, which is dependent on the sugar concentration, okay? So this is the kind of first clue that these clusters aren't necessarily empty, just lumps of particle. There's something else going on here. Okay, actually the rate at which this happens is reasonably in line with the change in solubility and diffusion coefficient. Okay, so what people haven't done previously is then actually take a close look at these clusters which you get at the end of compositional ripening. So this here is a cluster where the water is dyed, okay? And so in order to make the confectionery product, we'd like to trap the water permanently within a cluster. And this kind of makes it look like within this particle cluster there is water. Okay, this is a bit, we weren't that happy with this because obviously the dye can adsorb onto the particles and stuff. So we weren't completely convinced that this was water that we're looking at. And so we changed the whole system so that everything else was dyed except the water, and then looked at the kind of end state clusters. And you do indeed have these black regions corresponding to water. So it does look like in an appropriate solvent, you end up with these clusters, but the clusters basically end up with water trapped long-term within them, okay? Exactly how this works isn't immediately clear. Okay, so, yeah. Yeah, maybe I can stop that. No, okay, forget it. Okay, so yes, so it kind of we've got away from this explosion. We've created clusters, and the clusters make it look as though we're able to kind of trap water long term, which is basically what Mondelez wanted. I'm not sure, you know, how happy making this is, because you've got quite a lot of particles in there in order to trap a tiny amount of water. So it's not exactly what you'd want as a kind of design motif. Where we tried to go from, so this is even more edible, obviously. Where we tried to go from here, ultimately what we'd like to do is, we've got this kind of regime of exploding droplets, and we've got a regime of crumpled droplets which it contains some water, and we'd like to kind of be able to go between these states by modifying our system systematically. So we've tried various things, and we haven't really won yet, I would have to say. So one thing we did was to kind of copy something Tony Dinsmore did back before the dawn of time. And you can basically use toluene to get the steric hairs to collapse on PMMA, and you can basically sinter PMMA together. Okay, and so that was one route we went. And another route we went was to modify the attachment of the particles to the water-oil interface, okay? And the sad thing, I'll show you one example, is that each case brings up kind of new phenomenology, and it's not kind of intermediate between the other two, okay? So when you, so you can put in toluene, and you can collapse the hairs, and then you get, you basically get sticky PMMA, which is sintered together, as it were, by van der Waals interactions. Okay, and in order to make the droplets, you make an emulsion, you add the toluene, you stick the particles together, but then when you bring in the other droplets, you don't want the toluene there anymore, so you wash it away. And the emulsions look okay. I mean, they've been beaten up quite a lot because you've changed the continuous phase twice while making the sample. So they kind of look a little bit kind of dimpled, but they're not too bad. And so they kind of collapse like our dodecane. They kind of almost do an intermediate thing. So the pure water droplets lose their internal phase, but rather than either exploding or crumpling, they then sort of crack and they just get stuck together in sheets. So that's kind of where we end up. So that's water droplets kind of shrinking, they crumple, and then ultimately I've got another picture which shows it more clearly. You basically end up with these kind of sheets of particles all stuck together. There's no water anywhere, it's all gone, but the particles don't redisperse as in the exploding case. So this is slightly a work in progress, and we've been sort of then using interfacial rheology to kind of explore the interfaces in these sorts of systems as we go along. Okay, I think I'm going to leave it there. But half my talk was inspired by lunch, and the other half was an attempt to kind of play with trapping water inside chocolate with kind of mixed levels of success. Okay, so this is a photo of a jolly we sent some of the PhD students on at the end of the pandemic, but it happens to feature Raj, who worked on the ripening project, and Yu Chen, who worked on the foams project, and then there's other collaborators from Edinburgh and beyond. So thank you for listening.
Thank you very much. Questions, please.
Yes, yeah. Yeah. Yeah, right. So then it basically then stratifies into you know, the aqueous foam at the bottom and the oil foam on the top. And so basically the two, when you haven't got droplets of one in a continuous phase, the kind of phase separated, there's no continuous phase. You've kind of got the two phases wrapped around each other in the kind of bicontinuous thing, but it doesn't last that long. You know, the phases start coming apart. Yes, both phases still have air bubbles, yeah. Sorry, he's in charge. Yeah. Yeah, between the two liquids, yeah. No, we haven't. And this, so you use Ludox silica, and I'm afraid I didn't go that high up market. So this is then fumed silica, right? So it's a bit messy, and you tend to have these kind of clumps of particles. So there probably are, so you make a foam, but then there are particles which are probably free to be knocked off, you know, as you shake this system up, and it appears that's what's happening. So a nice way to take that foam project would be to move towards monolayers of model particles and see what happens if there's nothing extra that you can get out of the system. Yeah. No. Yeah. 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 So we don't fully understand the colloidal science in tributylene. I'm not it may be charge related, but we don't deliberately put salt in the system, although obviously, and it's PMMA, so there's not immediately, you know, obviously counterions knocking around. But then what we find, so I showed you when we initially make droplets in tributylene, there's particle clusters in the tributylene. So the PMMA doesn't disperse perfectly. Okay, you may now explain to me what's going on here, but the PMMA doesn't start clustering within tributylene unless there's water droplets around. Okay, if you just disperse the PMMA in tributylene, it's kind of fine, but as soon as you start emulsifying, then the particles which aren't emulsified start to cluster. And if there's any capillary bridges, they're way too small to see on a confocal. We can't see any traces of water anywhere. But yeah, the colloidal science of the tributylene isn't totally under control. But I'm not sure it's charged. But maybe you'll tell me otherwise. Yes, yeah. So what I'm imagining is happening, so you've got this water on the inside and you've got oil on the outside, and the particles are stuck on the surface, kind of blocking shared area between the two liquids. And it seems like the compositional gradient is enough to finally kind of rip the water interface away from the PMMA, you know, so then you finally get the interface pulled away and then the particles go. Okay, so that's the kind of picture is, yeah, that's slightly what, yeah. Yeah. Oh, yeah. So then this is probably for cake frosting. I think this would be a little on the soft side. I think we're more towards the kind of softish, beginning to slightly be solid like yogurt kind of end of things. It's kind of yogurty somewhere between Greek and flowable, right? So I think it's possibly, yeah. So to get a cake frosting that you'd never want to eat, we're not quite there yet. Okay, that would need tuning up a little bit more. No, we haven't done that. I mean, the droplets are quite large as it stands, and no, we haven't. Yeah, we haven't got that far. Yeah. Yeah, right. So yeah, I mean, another route to go would be to say, okay, we're mixing an aqueous foam and oil foam. We need to do something about this interface, and then put something specific in to go after the oil aqueous interface, which could be diblock copolymers. But yeah, no, I certainly haven't done that. I don't think. Yeah, the other people working with aqueous oil foams are doing that kind of thing either, I don't think. Yeah.
One final question.
Yeah, we haven't, I mean, yes, I would have thought there is. I mean, you've got to kind of get in this zone where you don't destroy the bubbles, but you mix things as best you can, right? And we did, with other ideas in mind, we did look at trying to destroy or abuse these things afterwards, and we were kind of doing things with a windmill with a ball bearing in the sample, and looking at them as they dried, and I can't remember what else we did. Various other silly things, and you can break them up a bit more. You just have to, if you overdo it, you destroy the bubbles quite quickly also. So that's yeah.
Thanks, Paul.